the scary thing is, the really scary thing is that drug resistance can arise even faster. All right? You don't need this accumulation of different mutations that might take days in this case, and days under perfect conditions. Okay? Horizontal gene transfer is when DNA is passed directly from one organism to another. That DNA can carry resistance to one or several different antibiotics. So this is as if normally you only give your genes to your offspring, to your children. But it's as if here are some genes, I hand them over to you. And that's what bacteria can do. Now, the first way, there are three ways it can work. The first way is called transformation. Now, transformation was discovered in the 1920s in a very famous experiment by Griffith. What he did, he had 2 strains of strep pneumoniae. And this was lethal to mice. All right? So he had 2 variants, though. One was called smooth, and that's how it looked on an agar plate. It looked smooth. And that one, if you injected the mouse died. But the rough strain that was bumpy on the plate, if you injected it in a mouse, the mouse was fine. It was, we say it's avirulent or non-virulent. It can't cause disease. Well, if you took the smooth strain and you heated it up, you killed it, and then inject the mouse is fine. But if you mixed the heat-killed strain with the rough strain and injected in the same mouse at the same time, the mouse died. And he called this the transforming principle. He didn't know what it was. He didn't know about DNA. He didn't know anything at that time. He just knew that somehow these dead cells were giving something to the rough cells. Questions about that? So this, of course, was DNA that was being transferred. So in 1944, they were able to show that it was DNA that converted the rough cells to smooth ones that could cause disease. And DNA later was shown to be the genetic material of viruses in 1952. And this led to us now knowing that DNA is what contains genes, which produces all the phenotypes of people and organisms. So what transformation is when you have a piece of DNA, and it can be a circle or it can be a linear piece of DNA. Some cells actively take up DNA from their environment, and they either keep it as a circle, as a plasmid, or they actually recombine or integrate it into the chromosome. Some bacteria seem to like to do this, and they take up DNA quite easily. Other bacteria can do it kind of by accident or by mistake, or they can be treated under certain conditions to be able to take it up. In the lab, we often use E. coli, and we can kind of trick it into doing this, but it doesn't normally do this. Bacteria that can take up DNA are called competent. They're competent to take up DNA. That free DNA, especially the linear DNA, may be degraded or introduced into the bacterial chromosome. So now I have a question for you again. Why are some cells naturally competent? Why would they do this? And you can think for 30 seconds or discuss. Well, there are 2 big reasons. The first reason is they can use this for food. So some strains or some bacteria pick up the DNA and they chop it up and they use it to make their own DNA. Right? It's food, basically. So they actually can use that as nutrients. Some people think, but this is less proven, that some bacteria do this because they want to increase their genetic variability. That would be a good thing to pick up genes in case one of them might be a good gene. That is very difficult to prove, whether or not that's true or not. And there's a lot of debate about whether or not that's true. But certainly the food is easy to see why they would do that. Is that clear? Okay. So these circles that I've been drawing for the last week are plasmids. And plasmids aren't just a circle of DNA. They are a circle of DNA, but they also are autonomously replicating. What that means is they have sequences that allow them to copy themselves along with the bacterial DNA. So when the bacteria is growing and dividing, it will make a copy of itself so that both daughter cells get a copy of the plasmid. Some plasmids have only one copy of the plasmid per cell, and some plasmids have lots and lots of copies. They could have 100 copies in the cell. They're usually smaller than I've drawn here, but yeah. So plasmids are replicating alongside the chromosome, which is why they keep them. Because otherwise, if this cell divided, one cell wouldn't get one. And the reason why plasmids are so important to us is that they are very common and they can carry genes for antibiotic resistance. So if a cell picks up a plasmid that carries a gene for resistance, for example, one of those EFFLUX pumps, it instantly becomes resistant to everything that EFFLUX pump, or not instantly, but soon becomes resistant to everything that EFFLUX pump makes it resistant against. In addition, they can carry 2, 3, 4 different antibiotic resistance genes and become resistant to many at the same time. This is one thing that happens a lot in the biofilm, we think. Questions about that? Okay. So I just want to make a comment. I won't go into detail on this, but where plasmids come from? They're like these magic things I'm talking about. Well, plasmids evolve from other mobile genetic elements, such as bacterial viruses, that we're going to talk about a little later, and transposons. Transposons are genes that can hop around in the chromosome. So they usually derive from either transposons or bacterial viruses, as well as some bacterial genes. So that's where they evolved from. So transformation is when just naked DNA, boom, comes in. There is, in nature, a bigger problem is something called conjugation. And conjugation is a very common type of horizontal gene transfer. It can spread to unrelated cells, and it can cause a rapid spread of antibiotic resistance. So this also uses a plasmid, all right, but it's a slightly different plasmid than the circle I drew on the previous slide. This one encodes proteins. They allow it to make a pilus. And I mentioned pili last week. And this is a specific kind of pilus. And yeah, for different plasmids there are different pili, but for each type there's only one. And this plasmid is called conjugative plasmid. And this is the pilus that is used for conjugation. Here's a picture of what it looks like. These are two cells. And one of them has a pilus. It is quite long. And it can attach to another cell. What happens then? Here's the donor cell That's a cell with the conjugated plasmid, and it's resistant to some antibiotic, or many. And here is a nice antibiotic-sensitive cell. That pilus will grab hold of the sensitive one, and the pilus then retracts to bring the two cells very, very close together. And then this plasmid gets a little cut in it, a little nick. And the DNA on one strand then is unwound and transferred through from the donor cell to the recipient cell in this close connection region. Then the cells both end up with one circle, one strand of the DNA. They then replicate that so that now you've got two plasmids, one in each cell, and both cells are resistant to the antibiotic. This can happen, there are lots of kinds of these plasmids and different pili and things, but this could happen sometimes between cells that are closely related, like two E. coli cells, but it can also occur between very, very, very different types of bacteria. And this, so this becomes particularly scary with regard to a question you were asking about earlier, is that if you have nice, friendly bacteria that have one of these plasmids, if you later get a pathogen, you get something that causes disease, it can be transferred to the pathogen. So we do worry about antibiotic resistance genes even in cells that don't cause us harm, because there are ways they can transfer to pathogens. Is this clear? This is really fast, by the way, and very efficient. This can occur in minutes. All they have to do is be close enough. They just have to bump into each other. Boom. Okay? Okay. And now the last general way that DNA can be transferred between cells is something that uses these bacterial viruses I've talked about. These are known as bacteriophage or just phage, we often say. And this is a picture of probably the coolest looking phage we know about, where it has a head here on the top, which contains DNA. Some phage have RNA, but this one has DNA. And then it has this apparatus here, which is essentially a way of injecting its DNA into cells. The black parts here are protein, and the blue is DNA. This is known as T4. That's its name. So phage are viruses that infect only bacteria. I'm going to come back to how they work. Scientists estimate there are 10 to the 30th bacteria in the world, but there are 10 times the number of bacteriophage, is the estimate. So for every bacteria, there are 10 phages. And we have shockingly little knowledge about the diversity of phage. We know a lot about a few, and we know of many of them, but we don't know much at all compared to bacteria. So bacteriophage are all around you. You have them in your gut. They're in rivers. They're just everywhere. And usually, again, they don't cause any trouble. There's no problem. But they do cause trouble to bacteria. All right, here's the phage. I made it a little simpler here. What happens if a phage bumps into a bacteria? If it's a bacteria that has a specific receptor on its surface, the phage can attach to it. Phage are very specific. Unlike conjugation, which can be very promiscuous, phage often only will infect one type of bacteria. And they usually bind to some protein on the surface. So it's there on the surface, and then the phage DNA is injected into the bacterial cell using an apparatus like the one I showed, which is almost like a syringe that pushes the DNA into the cell. Then the DNA will start to replicate, and you make more copies of the DNA. You then start transcribing RNA from this DNA and making proteins. You make all the proteins, you've made all the DNA. What then happens is many phage are cruel, and they actually chop up the host DNA, so the bacteria's DNA. I don't know why it does this, because it's going to kill it in a few minutes anyway, so I don't really understand why it cuts it up, but it does. And it actually becomes important for us. And then what happens is they assemble new phages, and there can be around 100 phage per bacteria. Remember that these phage are very small. Then it will make proteins. It has made proteins, which cause the cell to lyse, to burst open. And then those phage come out, and they can't move, but they can diffuse in the area, in the water, for example, and infect again. A typical phage, as I said, might produce 100 new phage in as little as 20 minutes. So it's a pretty fast process. If I show you what it looks like for real here, again, this is an agar plate, and there are cells growing all over here. But on this plate were put approximately 100, 150 phage. And what has happened here, you see these little holes in the agar, what look like holes. What that is are dead cells. And what has happened is that a phage attached to a cell, it killed it, and made 100 new phage. It went out, it killed the 100 cells around it, and 100 cells around it, and 100 cells around that. And it produces this area of dead cells on the plate. Each kind of phage has a different appearance. Some of them look very tiny, some look very big. It depends how they grow and how fast. Okay, so two more things I want to tell you about phage. First is this. Phage can be bad indirectly. And the reason they can be bad indirectly is that they can promote a kind of horizontal gene transfer called transduction. And transduction occurs when a phage infects the cell, and I've just labeled some gene, gene Y. It infects, it kills, it's going through all its process. Occasionally, it will, by mistake, instead of putting its own DNA, which I've drawn blue here, in the new phage, it will accidentally package up some of the chromosomal bacterial DNA. And that's because it chopped it into little pieces that it could look around the same size as a phage DNA. And so by mistake, it puts bacterial DNA instead of phage DNA. Okay, so you have this mixture here. What happens then, if the blue one infects, the same thing will happen, the lyse and everything. But if this red one that carries bacterial DNA with gene Y on it infects the new cell, it can actually combine with the chromosome and put gene Y into a new bacteria. It doesn't kill the cell because there are no phage DNA here. All it's doing is delivering this DNA to a new cell. If this were an antibiotic resistance gene, this cell would now become antibiotic resistant. Right? However, I want to emphasize, the most common way resistance is spread is conjugation. This happens, but the most common way is conjugation. But the reason I'm telling you about this is something called, well, I'll come to that one second. I want to talk about two new approaches or alternative approaches to antibiotic resistance. You're going to hear more as the course goes along, and especially in the last class. But I want to point out two approaches right now. And the first of them, someone asked about last week, which is phage therapy. So why not, we've got these killer viruses, right? Why not we use those? Well, this was tested when phage were first discovered in the 1920s, or first really characterized. But they tried it and it got inconsistent results. Sometimes it seemed to help an infection, other times it didn't. That was probably due to a lack of understanding about specificity. Meaning, I said earlier that these phage infect specific bacteria. So if you give a phage against E. coli, but you're treating a patient that has Staph aureus infection, it's not going to work. And there wasn't very much understanding of that at that time. So, but then shortly after this, while they started working, if you read a book, a novel called Aerosmith, fantastic book, mostly a fantastic book, and old movie. It was written around late 1920s. And he becomes a researcher in the book, and he's actually studying whether or not phage could work against bacterial infection, which is, I thought that was amazing when I read that. Anyway, when antibiotics were discovered, though, right, and became easy to produce in the 1940s, we decided, okay, we have antibiotics, we don't need phage. That wasn't working so well, we move on to antibiotics. Now, though, this idea has been revitalized, and a lot of people have begun working on using phage against bacterial infections. For example, there is a clinical trial being done in the EU for patients that have burns to try to prevent bacterial infections by treating them with phage on the surface where their burn is, to try to prevent that. And that's ongoing now. Another approach that is being taken that was recently published in the last year or so has to do with two things I told you about. EFFLUX pumps, which you remember are these proteins or protein complexes that can pump out the antibiotic, and phage. What they did was they had a patient who had very bad infection, who was untreatable, basically. And they isolated a phage that bound directly to the EFFLUX pump. Okay? So they looked for that, they found it. And what that means is, here's the cell with the EFFLUX pump. This cell is sensitive to the phage. It will be killed. But it's resistant to antibiotics. If that cell mutates, it will lose the EFFLUX pump, so it becomes resistant to the phage, but now it's sensitive to the antibiotic. So what they tried was to treat the patient with both phage and antibiotics. So it had no escape, so to speak. They could attack on both sides. And this worked. It was one patient, one time, etc. So it's very new research. But it worked remarkably well to be able to use it as a supplement with antibiotics. And so this is the kind of thing people are thinking about to try to essentially use resistance against the bacteria. Questions about that? All right, and one last way in which people are thinking that someone actually mentioned. So here's a cell with the EFFLUX pump again, where it's pumping out the antibiotic. People are designing inhibitors directly for the EFFLUX pump. And what that will do, hopefully, this is again very new research, is by inhibiting the EFFLUX pump, you can resensitize the bacteria to the antibiotic. So you could treat with both an inhibitor of the EFFLUX pump and an antibiotic. And then we can get use out of old antibiotics that aren't so effective anymore. Okay, so my second to last slide is an advertisement for next week. We've talked about the spread of antibiotic resistance between bacteria, but I've been ignoring the spread of antibiotic resistance around the world or between people, etc. And that's because a lot of this can be covered next week, where you're going to have 2 lectures about the environment and antibiotic resistant development and spread in the environment, in animals, in people, and in nature. And then my last slide. If you are logging for microbiology news on an almost daily basis, you're welcome to follow my Twitter and Facebook feed. And right now, because so much is about antibiotic resistance, at least half of the microbiology news is about resistance.